Thank you so much for that nice introduction. I'm a little embarrassed how long it took me to worm my way on to the Center for Healthy Aging seminar schedule, but really happy to be here. It looks like a small enough group that I wish we were sitting around a table somewhere instead of on Zoom, but at some point we'll do that. I do think this is a pretty mixed group. Some of you know much of what I'm going to present. Others probably don't. And so my goal is to give you a little science, but if you get bored with the science, just hang in there and I will always give you kind of a bottom line to what we think our findings mean too. So hopefully it'll be a little something for everybody. So what I hope to do today is to kind of highlight some features of three sort of sets of studies that we've been working on over the years that are kind of geared towards this notion of, if we look at two good things, and by good, I mean they're good at helping us to extend the number of years that a species can live without the burden of chronic disease. That's what we call health span. So two things that can extend health span. If we add them together, does that always make a better thing? Or are there some things that we need to be concerned about? So interactions between health span extending interventions is the topic. I like to do thank yous up front, and then I'll also do them along the way. Always grateful to the folks who provide the lab with money to do the work that we're doing. Big thanks to Allie for organizing and always hosting the seminar so beautifully, and for squeezing me into the schedule. All of you for spending your lunch hour with me. And then over here on the right, the people that I have pictured are folks who have worked on some of the data that I am going to share with you today. So their work kind of features in today's presentation, but so many others have helped make and continue to help make the things that we do in the TRACD lab possible. So here's my plan. I'm going to start off with just a word or two on exercise and this notion of health span. And then after that, I'm going to sort of divide the rest of the time between looking at interactions with exercise and a class of compounds called NRF2 activators, or interaction with metformin, a drug that may sound familiar to some of you, and then interaction with another class of compounds called Sglt2 inhibitors. So thought I would just reiterate, make sure that I'm using terminology that everybody uses the same way. So when I talk about health span, I'm kind of using that in contrast to lifespan. So most people who study aging will agree that we're really not interested in extending lifespan if it's at the expense of the number of years that we really feel like we're not burdened by chronic diseases of aging. Meaning we don't want to extend this portion of this continuum where we're still alive, but we're really not enjoying our health at that time. So what we really want to do is extend the number of years that we live free of the burden of age-related chronic diseases, which should also help us to compress the number of years or the amount of time that we spend burdened with those diseases. So that's what I mean by health span. Easy to define, kind of hard to measure. So this is a really nice review paper that's actually getting pretty dated now, but I still like it. Huffman and colleagues kind of define these domains of health span that are considered particularly important for human health. And my dream is to be part of a group of collaborators that collectively we can take a really rigorous approach to measuring all of these domains of health span. And we're really getting very close to that, thanks to the Center for Healthy Aging bringing together people with such shared interests. So I'm pleased about that and grateful to the center. What I'm going to share with you today is going to mostly fall into these two domains of health span, metabolism or metabolic health, and musculoskeletal health, which is kind of falling into this domain. So I'll share with you a little bit of data focused on this notion of maintaining protein homeostasis, which you collapse together into the word proteostasis. That's always been one of my favorite things about science. You're allowed to make up words. So I'll show you some some data on protein synthesis, which is a really important component of this maintaining healthy proteins in our cells. I'll show you quite a bit of work focusing on mitochondria, which are the organelles in cells that help us to make the energy that we need to do everything. So it's a measure of metabolism. I'll show you just a little bit of data on glucose homeostasis, or how we handle glucose to reflect metabolic health. Every time we do a study, we try our very best to include some component of movement, whether that's exercise capacity, daily physical activity, maybe breaking it down into aspects of gait. And I'm very grateful to two collaborators in particular who are experts at measuring gait in preclinical and clinical models. So that's a fun thing to add to our studies. And with some of those collaborators, I've been able to gather some data on muscles, which is what we really study, is muscle aging. One of my colleagues studies joints. She's an expert in joint degeneration. And then another who's really focused on bones. So we're really starting to be able to cover this whole musculoskeletal aging quite thoroughly. So let's begin by telling you that whether or not exercise extends total lifespan is actually still somewhat debated, but there's very little debate that exercise extends the number of years that we spend healthy. And it does this by defending against what we know tend to be age-related declines in our cardiorespiratory fitness, how strong our muscles are. I've already mentioned this, our mitochondrial function, or energy metabolism. Also, there's age-related declines in balance and gait that exercise defends against those losses. And then over the past decade or so, we've really grown to appreciate the role that exercise can play in helping to prevent age-related decline in cognitive function. This is a really nice review by Drs. Rugsager and Booth that I've kind of adapted this figure from, where they review the literature and what is known about skeletal muscle contraction. They show a bicep here, but really large muscle contraction. So our legs contracting when we exercise is even a bigger driver of adaptations that probably result from systemic messages. So muscles contract and send out messages to literally every tissue in the body. So health span extending adaptations are very widespread, not just in the muscles that are doing the exercise. I have lots and lots of friends who study aging. Not all of them are focused on exercise. So I'm always quick to point out or to remind them that two of the best known adaptations to exercise, that is improvements in cardiorespiratory fitness and muscle strength, those two measures are the best predictors of all-cause mortality. So they're very important in the framework of aging. So hopefully that quick little first bullet convinced those of you who needed any convincing that exercise definitely extends health span. So with that as the backdrop, I'm going to move on to the three different categories of interaction of exercise with NRF2 activators, then metformin, then Sglt2 inhibitors, beginning with my favorite. And the reason NRF2 activators are my favorite is that I've been interested in this field called redox biology, or the biology of oxidative stress, since I was a student. And since that was a long time ago, it kind of gives me the ability to see how far we've come. So back when I was a student, we were really focused on these reactive oxygen molecules, pretty much as bad guys, that they're unstable. So all they really did was seek stability by stealing electrons from components of our cells. In doing so, causing damage to proteins, to our DNA, to lipid membranes of our cells. And it followed that if they were the bad guys, if we threw some antioxidant superheroes at them, then we could fix that problem. We could fix the problem of oxidative stress causing damage. We've come a long way because we know it's this is a much more nuanced process, that this so-called redox reactions or biology are important for many things to work properly in the cells. So I know that over the past year, we've all been thinking more about our immune systems than we probably did in the past. And we know that our innate immune system absolutely requires reactive oxygen species, or these unstable molecules in cells that can be made by virtually all cells. If they didn't have this ability, they wouldn't be able to fend off pathogens. But we also know that things that we're exposed to in the environment, like UV, air pollution, if we're a smoker or secondhand smoke, these all cause us to be exposed to even more reactive oxygen species, or ROS. Those studying aging have learned over the decades that aging itself, as well as age-related chronic disease, make it so that our cells are exposed more chronically to at least low levels of reactive oxygen species, and that this is at least a part of age-related chronic conditions. But we also know that movement, the exercise that we know is a health span extending intervention, causes production of reactive oxygen species. So again, over the decades, we've been really interested in this sort of emerging body of literature that's been addressing this idea of interaction between the antioxidant supplement superheroes that we thought might be part of the solution. And by this, I mean supplemental vitamin C and vitamin E beyond a healthy, well-balanced diet, and whether or not they interact with exercise. So some people believe that since exercise is a stimulus to produce reactive oxygen species, that antioxidant supplements might help to prevent any damage that might be caused by exercise. So I'm going to summarize it really quickly. You can see that those papers that are falling under the no, there's no unfavorable interactions between antioxidants and exercise. And those that conclude, yeah, it kind of looks like antioxidant supplements might interfere with exercise, they remained pretty balanced, pretty one for one, until fairly recently the scales are starting to tip towards, we should think about this before we really consume high doses of antioxidant supplements if we're hoping to gain lots of benefit from exercise. All right, this is about as sciencey as the presentation gets, and it's not very sciencey because I drew this picture. So this cartoon is meant to represent a cell. So here's your cell membrane. This part would be the outside of the cell. This part would be the inside. And we took kind of a fundamentally different approach to looking at interactions between redox balance and exercise by focusing on this transcription activator called NRF2. So it's not the only thing that regulates transcription of genes. It's just one thing. It's the one we're most interested in. And it sits in the cytosol, the liquid portion of cells, bound to its protein partner, KEAP1. And while it's bound to KEAP1, it's not really doing much. In fact, KEAP1 will take it to this fancy little machinery called the proteasome that's responsible for chewing up degrading proteins to keep them turning over. Normal process. It also makes it so that NRF2 only hangs around in cells for 15 to 30 minutes in the absence of stress. Now, when there's cell stress that can be sensed, then NRF2 is released from that partnership with KEAP1. It goes into the nucleus of cells. It finds sequences in our DNA that it recognizes. And when it does that, it regulates transcription of over 400 genes that collectively help to protect cells. So this might be a new term for some of you. I would call most of the genes that NRF2 regulates important players in adaptive homeostasis. So adaptation to the stresses that the cells just saw so that cells can return to normal. Homeostasis, adaptive homeostasis. So here's our fundamentally different approach to asking essentially the same question that folks had been asking about supplementing with vitamin C and E and exercise, and do they interact favorably or unfavorably. We hypothesized that treatment with vitamin C supplements would inhibit, in particular, important muscle mitochondrial adaptations to exercise, but that treatment with NRF2 activators, things that activate that transcription factor I just introduced you to, would permit mitochondrial adaptations to exercise. So this set of studies I'm going to show you first was with a, excuse me, a laboratory rat model. Rats like to run, and like rats, one of the best known adaptations to exercise is in the muscles, we make more mitochondria. So mitochondria are important for making the ATP, or the energy that we need to keep moving. And with exercise, we make more of them. So that's why these hypotheses are focused on muscle mitochondrial adaptations. So here's what we did. We took 3 groups of rats that liked to run, and we either kept them on a control diet, or we supplemented their control diet with essentially equal concentrations of vitamin C as some of those studies in that big long list I just showed you. Or we supplemented their diet with plant compounds that we can talk about later that activate that transcription factor NRF2 that I introduced you to. And then we had one more comparison group, rats that we allowed to stay lazy. We locked their wheels in their cages so they couldn't run, and they just stayed on a regular diet too. And one of our primary measurements is the measurement of new mitochondria that are synthesized in muscle so that they can maintain a healthier metabolism and greater energetics and metabolic health. All right, you have to bear with me on a few histograms if you hate them, and I'll get to a bottom line. But what we're measuring here, again, is in muscle that helps the rats move, and we're focusing on new mitochondria that are being made. So that's what's on the y-axis here, new mitochondrial proteins that were synthesized in muscle. So focusing on mitochondria. If we compare the rats that had to stay sedentary because we locked their wheels to the ones that ran like crazy every night, they're nocturnal, on their wheels, but ate a controlled diet, this is what we would expect to see: exercise-induced synthesis of new mitochondrial proteins in the muscle. However, if they had lots of vitamin C in addition to their diet, that completely prevented the exercise-induced increase in mitochondrial proteins in their muscles. However, if their diets were supplemented with the NRF2 activator, this was permissive. They still were able to have an exercise-induced mitochondrial adaptation. So again, this does not mean that they took that this diet and this diet have no vitamin C. This is supplemental. And in addition to that, here's a little party tip for you to share at your next gathering. Rats actually synthesize their own vitamin C. So that's why they're not a perfect model for human health. We all have to get vitamin C in our diet, and they're able to make it. So a little bit of a different model. If you really like these data, you can move on to a different set of proteins that are in the cytosol, so not in the mitochondria, they're outside the mitochondria. Saw the very same thing, that vitamin C interfered, NRF2 activators didn't. Myofibrillar proteins are the ones that help muscles to contract. So these are the contractile proteins. And I think this is really interesting. Vitamin C did not interfere with exercise-induced increases in our contractile proteins. What this suggests is that contractile protein adaptations to exercise aren't nearly as redox sensitive. So it's another little fun finding from this study that we think the conclusions here are that vitamin C supplements probably do have a high likelihood of interfering with some of the beneficial adaptations to exercise, specifically endurance exercise, and more specifically in the mitochondria. But these NRF2 activators are permissive, so they can turn on protective mechanisms, including antioxidants, in the cells without interfering with exercise adaptations. All right, this isn't my work. This is another really nice topical review paper. If you're interested in this, you might look it up. A few years old, but still very good. And what Drs. Davies and Pomato did was kind of summarize the literature focused on a whole bunch of different stresses that cells are exposed to. So far, we've been talking about oxidative stress and exercise as a stress, but they looked at thermal stress, energy stress, osmotic and mechanical, even emotional stresses. And they looked across experimental model systems, including cells in dishes, lower organisms, rats and mice, like the models I just showed you, and of course, people, which is what we're ultimately geared towards improving aging in. And they looked at these mechanisms of adapting to stresses to reestablish homeostasis. And the literature concludes that with youth, we have a really wide range of ability to adapt and reestablish homeostasis after a stress. But with aging, this range kind of gets compressed and we have fewer mechanisms to adapt and reestablish homeostasis. And remember, when I first introduced you to NRF2, I said that NRF2 plays a big role in these mechanisms or tools for adapting and reestablishing homeostasis. So NRF2 may become even more important with advancing age. So now I'm going to show you the only unpublished data. So these are pretty new data. Maureen Walsh, who's on this call, is part of collecting some of these data. So is Dr. Musey. Both of these talented individuals have moved on. And we did this work with some collaborators where we used a different preclinical or laboratory model now that we're all fully in love with. They're adorable and lovely to work with. This strain of guinea pig is called a Hartley guinea pig. And what's unique about them is that they develop joint degeneration that's like our aging related joint degeneration. I call it primary or spontaneous osteoarthritis because it's not in response to an injury. It just develops spontaneously, starting at a really young age, 3 to 4 months for these animals. Rob and Maureen were part of the team that led some studies showing that along with joint degeneration, their skeletal muscle starts to decline. And by the end of the end stage of their osteoarthritis, which is 18 months to 2 years old, they're at pretty advanced stages of the disease, their skeletal muscle starts to resemble muscle that's very similar to aging humans as well. So overall, this is a really nice model of overall or systemic musculoskeletal decline. So we use this model to test the hypothesis that treatment with this NRF2 activator that I've introduced you to, that's what this guinea pig is getting treated with right now, or a placebo would slow down the progression of this musculoskeletal decline, focusing on joint degeneration and skeletal muscle. All right, another histogram, but you already have been introduced to data focusing on mitochondria. So this is kind of just a fancy way of showing how muscle mitochondria work. On the y-axis is a measure of one of their most important jobs, consuming oxygen. Lots of bars going on here. The green ones are the NRF2 activator treated ones. The controls just got placebo. We looked at both males and females, and we did this at 2 different ages. So a few things to point out here. Like humans, from 5 to 15 months, if you compare the black bars, muscle mitochondrial function declines. Also potentially like humans, though these data aren't conclusive, males and females have different muscle mitochondrial activity, even at a younger age. So now let's focus on what the treatment did. So I pointed out this age-related decline, and in the males, treatment with the NRF2 activator prevented that age-related decline in muscle mitochondrial function. If we look at the females, remember the females have a lower muscle mitochondrial function even at a younger age than males, and the NRF2 activator essentially made them look like males. So they were no different in terms of muscle mitochondrial function than males. Unfortunately, in the females, it did not prevent the decline in muscle mitochondrial function with age. So we've got lots of questions to continue to pursue here, but also quite a bit of promise for improving muscle energetics with NRF2 activators. Same study, different part of the musculoskeletal system. And this work was under the guidance of Dr. Kelly Santangelo and Kendra Andre, who are experts in osteoarthritis. And that's what you're looking at here. I am going to simplify these a whole lot. This is a knee joint of the guinea pigs that were treated with the placebo versus the ones that were treated with a NRF2 activator. And if you just concentrate on the intensity of the purplish blue stain, that shows you components of the cartilage. So the more intense the stain, the less degeneration has happened in that joint cartilage. So much less degeneration in the NRF2 than in the vehicle. You're going to like this. Oops. There we go. So these kiddos like to run on a treadmill, and we can measure aspects of their gait. And I cannot turn off the sound there. And what we found was that gait, aspects of their gait improved with NRF2 activator treatment. And Dr. Christian Putlitz helped us to show that the bones also became stronger only in the females and not the males. We did this with a method where we apply stress to the bones, and the more stress that it takes for the bone to break, the stronger the bones. So I'm going to summarize that first set of studies by saying that both NRF2 activators and exercise protect against decreases in health span, and there's no evidence of unfavorable interactions. We're so excited about these data that we're already doing a small clinical trial in human participants with osteoarthritis. And we've proposed it in companion dogs who are just as susceptible to OA as we are, but we haven't gotten funding for that yet. So the next two sets of data are a little bit smaller, so the end is in sight. Now we're going to look at an interaction between a very widely utilized drug called metformin and exercise. So we've got decades and decades of safety and efficacy data of metformin for its primary diagnosis, which is treatment of type 2 diabetes. It's a safe drug. It's good at bringing down blood sugars. And this is a well-known study published a long time ago now that came from the Diabetes Prevention Program, where they compared people treated with a placebo over 4 years to people treated with metformin or interventions to encourage lifestyle changes, including increasing physical activity and changing dietary habits. And their outcome measure was cumulative incidence of diabetes. And so what you can see is that in comparison to placebo, both metformin and lifestyle interventions decreased incidence of diabetes, which is another way to say that both are capable of increasing aspects of health span. So Dr. Adam Konopka was a postdoc in my lab when he wanted to address this hypothesis that metformin will accentuate the positive adaptations to exercise. And we would see this by observing greater improvements in skeletal muscle mitochondrial function, which you're now an expert in, insulin sensitivity or glucose tolerance, and whole body oxygen consumption or cardiorespiratory fitness. So this was a pretty simple study in theory. And what we did was recruit adults who were over the age of 55 that had at least one risk factor for type 2 diabetes in this list here. And this was a study design where we did some testing before the intervention and the same testing after the intervention. It included something called an oral glucose tolerance test to see how well they tolerate glucose, how insulin sensitive they are. We measured their whole body oxygen consumption or their cardiorespiratory fitness with VO2 max. And then we looked at muscle energetics by looking at mitochondrial function. Then these previously sedentary adults engaged in 12 weeks of supervised aerobic exercise training, during which time they were either taking a placebo or they were taking metformin. And then we repeated all of the testing. And the metformin dose we used would be considered a clinical dose for treating type 2 diabetes. I'm going to show you 3 pieces of data. 2 are on this slide, 1 is on the next. The first one, all of them, I'm going to show you as a change or a delta, which means it represents how much this measure changed from pre or before exercise training to post exercise training. So this first one is the glucose tolerance test, where we see how well they take up sugar, take it out of circulation into their tissues. And so anything above the black line represents an improvement. Anything below represents a decrement. So as you can see that nearly everybody that was taking placebo either stayed the same or were able to increase glucose uptake or improve insulin sensitivity. What should leap off the screen in the green circles is that those taking metformin, about 50% of them actually had a decrease in their insulin sensitivity, and the other half an increase, with a number of them clustered around no change. So this is not what we would expect from exercise training. We would expect exercise training to improve insulin sensitivity. So there's obviously an interaction here with metformin. This is cardiorespiratory fitness, a change in VO2 max with exercise training. And you can see that in comparison to the gains that the placebo-treated group made, metformin enjoyed only about half of the improvements of the placebo group in cardiorespiratory fitness. This looks complicated, but it's not. You've already seen it before. This is the fancy way of measuring muscle energetics, where we're looking at how much oxygen uptake there is into skeletal muscle, how well the mitochondria work. And again, showing this as a delta, an improvement from pre to post training, which is what we would expect and what you see in the black bars. So at every different measure of this protocol, which if you're super interested in it, we can talk about it later, the bars mean there was an exercise-induced improvement. If you look at the green bars, you can see that metformin essentially took away or prevented those exercise-induced improvements in muscle mitochondrial function. So the summary for this section is that contrary to our hypothesis, metformin really did not accentuate the effects of exercise training on glucose tolerance, insulin sensitivity, or skeletal muscle mitochondrial function. I've underlined this cardiovascular disease risk finding because I didn't show you these data. However, our data are supportive of a series of studies that our department head, Dr. Barry Braun, carried out before he came to CSU, showing that metformin definitely does not accentuate exercise-induced improvements or decreases in cardiovascular disease risk. And both his work and ours suggest that metformin can blunt some of the beneficial adaptations to exercise. So in my opinion, in the setting of aging, metformin, the cost-benefit ratio of metformin needs to be considered. There's still quite a bit of work to be done to understand this full cost-benefit ratio, but particularly in people who are highly likely to adopt and adhere to an aerobic exercise regimen, we need to consider whether there's an alternative to metformin, or at least put a plan in place to measure if they're getting everything they can out of their exercise training, because it's hard work and you want to get something out of it. In the meantime, we kind of shifted our focus to another class of drugs that can be used to treat type 2 diabetes. They're called Sglt2 inhibitors. So I had to learn what these were. I figured there might be some people in the group who don't know what they are either. So Sglt2 stands for sodium glucose cotransporter 2. And these transporters sit in the kidneys, in kind of the filtration units, the workhorses of the kidneys. So they sit here. The whole job of the filtration units in our kidneys are to filter our blood. They do about 180 liters of plasma filtration in adult human per day, which equates in a normal glycemic person to about 160 to 180 grams of glucose a day. And what these transporters do is to reabsorb about 90% of that glucose back into the bloodstream. So it should make sense to you that if we block those transporters with a drug, then they're not going to reabsorb that 90% of the glucose, and we're going to lose more of that sugar in the urine. And that's the mechanism of action, primary mechanism of action of these drugs, why they're so good at decreasing blood glucose. Right, here's the last study, the pièce de résistance. Melissa Linden was a postdoc in my lab pretty recently, and she was interested in addressing the hypothesis that when we use an SGLT2 inhibitor. She used one called canagliflozin, which is just too hard for me to say, so we're going to call it an SGLT2 inhibitor. When it's combined with exercise, there would be further improvements in glycemic or blood sugar control compared to exercise alone or to drug treatment alone. And in comparison to metformin, that I just showed you our studies on metformin, she hypothesized that SGLT2 treatment would not attenuate or interfere with any of the exercise adaptations. We're going back to the running rats or the sedentary rat model. You're already familiar with them. The only thing different here is that to make rats have hyperglycemia or to make them start developing type 2 diabetes, we have to feed them a high-fat diet. Otherwise, they're pretty resistant to the disease. This is a bunch of data all looking at the same thing. This is the glucose tolerance test, where we give them a bolus of glucose, just like you'd have done in the lab if your doctor ordered it. And then over 2 hours, we measure their rise in blood sugars and their decline. And then we measure the area under this curve. So the higher, the bigger the area under this curve, the more glucose intolerant they are. So that's what this graph is showing. I think it's easier to show the data here. So first, if we compare a regular diet, not high-fat diet, in a completely untreated rat to the rats that got the high-fat diet, so we made them essentially diabetic, you can see we were successful, and there's an increase in glucose intolerance. If we exercised them, we prevented that increase in that area under the curve, meaning they remained more sensitive to insulin or tolerant of glucose. If we put them on the SGLT2 inhibitor, but let them stay sedentary, that also improved their glycemic control. And then here's kind of the big finding. If we combine those two treatments, both drugs and exercise, that gave us the best improvement in glucose tolerance, better than either exercise or the drug alone. So the take-home message here is that our studies in animals showed that the SGLT2 inhibitor canagliflozin plus exercise training led to further improvements in glucose tolerance compared to either of those treatments all by themselves. Last data. So now we're only looking at the rats that ran. And so they either had the placebo treatment or the drug treatment. And you could see that by the end of the study, there weren't that many rats that really wanted to run anymore. We've repeated this study with an equally small sample size, but we've now shown the same finding twice. In this graph, while they're running at a pretty low intensity, we measure their respiratory exchange ratio, and you can see that the ones treated with the drug have a lower RER. The meaning of this is that this suggests they, during exercise, had a greater reliance on fat for energy. Also running on the treadmill, first at a low intensity, and then we pop them up to a higher intensity. And then all we're doing is seeing how far can they run till they are too tired to run anymore. And you can see that the ones treated with the drug ran longer. It's actually about twice as long, but certainly more variable than the ones that were treated with the vehicle. So here's the take-home message. While metformin interfered with adaptations to exercise, it looks like this SGLT2 inhibitor has a permissive or maybe even an additive interaction with exercise training. So very different findings from those two groups of drugs used to treat type 2 diabetes. Again, a lot of these are preclinical studies, not yet in people, but I think there's a lot of questions to be asked about whether SGLT2 inhibitors may be a good alternative to metformin, particularly in people who are very likely to adhere to an exercise as a medicine, a therapeutic exercise program. That is all I've got, besides some concluding thoughts that I hope I've convinced you that good plus good doesn't always equal better, that it's important to look at interactions between health span extending interventions. And that's something we'd like to do moving forward. We'd also like to gather as a full lab group. We haven't all been in the same room for over a year. This is the current group, 1, 2, 3, 4 undergrads in the lab, a master's student, and a very talented senior scientist. And all of them work very hard and help to make Track D Lab a great place to be. So if you have any questions, I will do my best to answer them. Thank you for listening.
Thank you, Karyn. If anybody has any questions, feel free to pop them in the chat or unmute and ask them in person.
Could you put that second to last slide on that gave us a summary of your report?
Absolutely, that one?
Various studies. That way I can study it and
absolutely
my takeaway. So basically, okay, I'm just trying to figure this out. I'm a non-science aging senior with osteoarthritis, and I don't know if I should take away, don't do vitamin C supplements. Do you need meds for your pre-diabetes? So I don't know.
Absolutely.
I don't know what to take away.
Let me boil down the takeaways a little bit for you. So the NRF2 activators were, those studies were mostly focused on exercising, and that NRF2 activators, which you actually can buy some commercially, even some of the ones that we used for the studies are available commercially, that they may be a better way to go in terms of a nutritional supplement than adding more vitamin C and vitamin E to your already well-balanced, healthy diet. So the caveat there is that if you don't eat a diet that's sufficient in vitamin C and vitamin E, that's the first thing to figure out. So all of these are kind of applicable only if you assume a well-balanced diet. It doesn't seem like vitamin C and vitamin E are a great combination with endurance exercise training if you want to gain maximal benefits from your endurance exercise. NRF2 activators, preclinically, so what I mean by that is that's still in an animal model, seem to have a lot of potential for both osteoarthritis, slowing down joint degeneration, and helping muscle to work better with aging, having it better energetics. We're just now starting to translate or move those studies to humans, older humans with joint degeneration or mobility impairment. So I'm going to ask you to stay tuned, and hopefully in a year or so, I'll have some data in humans that I can share. Metformin, the data from science, from scientific studies are mounting that this very good drug for treating type 2 diabetes. There's nothing arguing that it's a very good drug for treating type 2 diabetes. But if you are hoping to gain maximal benefits from exercise, metformin may get in the way. So that's something you'd want to talk to your doctor about before demanding big changes in your pharmacological, your drug plan for treating type 2 diabetes, but it does seem to interfere or interact with exercise. The newest set of data are these new hard to say drugs, the gliflozins, canagliflozin, dapagliflozin are two of the three drugs that are approved for use in type 2 diabetes in the United States. There's more approved for use in other countries. And in our animal studies, these drugs seem like they might not interfere the way that metformin does. In fact, it looked like they added to some of the exercise benefits that we saw in our animal model. Definitely still needs to be translated to a human population before you really adopt any of the take-home messages from that one. Did that help?
Yes, definitely. Thank you.
Good, absolutely.
All right, we got a couple in the chat. You made a convincing case that some common medications can interfere with the beneficial effects of exercise. Is there any reason to be concerned about the converse, that doing aerobic exercise could interfere with the beneficial effects of the medications?
Absolutely. And I actually, if you want me to get on my soapbox for a little while, I think that our primary federal funding agencies don't don't invest enough in looking at interactions overall, drug-drug interactions, drug lifestyle interactions going both directions. So I wish that I could convince everybody that this is an important thing to focus on. It is, if we look at just the type 2 diabetes drugs, like the ones we've looked at, metformin and Sglt2 inhibitors, their whole goal as a class of drugs is to improve glucose tolerance, right? To make us have better glucose homeostasis in our blood. Exercise does the same thing. So it's difficult without having this well-controlled, here's the drug, here's exercise, here's both. If we only had the combination treatment, there's no way we would have really known whether exercise was interfering with the drug or the drug was interfering with exercise. But we could still see improvements in the mono treatments for both. I didn't show you metformin only data because we didn't have a group without exercise in the study that I showed you. So I do think certainly with other drugs that are geared towards treating other conditions, we should exercise can change the metabolism of drugs. So it may change clearance, it may change what metabolites or byproducts of those drugs are available. So absolutely those are valid and important interactions to consider as well. We did not with these studies.
In the Konopka study, there was a relationship between delta insulin sensitivity and delta VO2 max, where the favorable responders and non-favorable responders to exercise combined with metformin consistent across multiple outcomes.
I think what you're asking is, could I look at this dot, this person, and predict whether they had a good VO2 max response or a bad VO2 max response? And if I got your question right, the answer is no. So we looked at all of our outcomes to see, could we predict in particular who responded unfavorably versus favorably in terms of insulin sensitivity? Meaning, if they didn't get the gains in VO2 max, did they also fall below the insulin sensitivity line here? The answer is no. I think we need a whole nother study geared towards what is the best predictor of who's going to respond favorably, maybe have no interactions with exercise and metformin, and who's more likely to have an interaction. That is an excellent question.
Is there a commercial NRF2 activator supplement we can purchase now?
There are, and I'm certainly not promoting anybody's product. The two that I know about, one is made by a company called LifeVantage. It's called Protandim. And another is made by a company called Pathways Bioscience. And that product is called PB125. And I suspect there are, well, I know there are others. In fact, something that you may eat in your diet every day is also a NRF2 activator. For example, broccoli. So broccoli has a compound that kind of makes it smell bad when you cook it, called sulforaphane. Sulforaphane is a NRF2 activator. Indian food, if you like Indian food, you're probably taking in a compound called curcumin in the spice turmeric. Curcumin's a NRF2 activator. So lots of different phytochemicals, plant-derived chemicals, things that have, well, if you want me to get down to the chemistry of it, that have phenolic rings in them, a lot of them are good NRF2 activators. What's unique about the ones that we used is that they're combinations of phytochemicals that each of them is at a very, very, very low concentration, that if you used just that one plant of the combination, you get no NRF2 activation. When you put them all together, or very little. If you put them all together, they synergize, they work together to be very powerful NRF2 activators. So I hope that answered your question. All comes back to the well-balanced diet. Get your vegetables in.
I think that's always good advice. All right. Well, if no one has any more questions, we'll go ahead and call it for the day. Thank you so much, Dr. Hamilton, for joining us.
My pleasure.